In the previous section, we ended talking about exons and introns. Introns are these intervening sequences that are spliced out by enzymes before the messenger RNA transcript is transported outside the nucleus, whereas the exons, the exon 1, the exon 2, the exon 3 remaining at the bottom are going to be those express sequences that will be translated into a protein. Thus, once the messenger RNA is transported outside the nucleus, that ends the process of transcription. So at this point, transcription has occurred. Next, we will move out into the cytoplasm into translation. Translation is going to convert the nucleotide language, taking that DNA template into the mRNA, messenger RNA template, and going to convert it into amino acids into the language of proteins. Amino acids are the building blocks of proteins. So translation will occur in the cytoplasm on the ribosomes. Our genetic code is a term that describes converting these DNA-based sequences into an amino acid sequence, in other words, into a protein. Codon is a term, and these are the words in the genetic code. They're going to be in a 3-base sequence, and they're going to translate into one specific amino acid. For example, UUC on the messenger RNA is going to specify the amino acid phenylalanine, which we will see in our amino acid table in a moment. There's 3 stop codons, which will be either UAA, UAG, or UGA, which will signal the end of the translation of a particular protein. They do not code for an amino acid. So once one of these 3 sequences are incurred, the translation of that particular protein will cease. This is an amino acid table. If you look along the left side, you see a U. Along the second base, up at the top, you see a U. And along the third base on the right, you see a U. That UUU codes for the amino acid phenylalanine. You will also see that UUC right below it codes for phenylalanine. If you look down further, if we look under the CUU, you see it codes for leucine, as does CUC, or CUA, or CUG. There's 20 amino acids, but there's 64 possible codons. So these multiple codons, these 3 sequence codes, code for the same amino acid. So if we were translating a protein, and for example, there was an error occurred, and it should have been CUU, but it ended up being CUC when it was transcription occurred, we would still make the same amino acid. We would still make the same amino acid would be used and the same protein. Thus, having these variations by being 20 amino acids and 64 codons, this allows us a greater variability to make the correct protein were an error to occur. Transfer RNA or tRNA, it's going to convert our genetic message carried by the messenger RNA into a particular sequence of amino acids. tRNA is a specific type of RNA that's going to be responsible for delivering amino acids one at a time in an ordered sequence directed by the code on the messenger RNA. It recognizes both the codon on the messenger RNA and the amino acids specified by that codon, that three-letter sequence. Specific enzymes ensure that tRNA binds with the correct amino acid. So it's going to ferry these amino acids to the correct location along the mRNA strand. Anticodon is the term for this three-letter sequences that's located on the transfer RNA. This 3-base sequence on the tRNA is complementary to the messenger RNA codon. It binds to a codon on messenger RNA, and the specific amino acid is added to the growing polypeptide chain on the ribosome. This shows the anticodon. So you see the amino acid. In this instance, it gives on the messenger RNA, that 3-base sequence is UUC. That would be the UUC would be your codon. Your codon is your 3-base sequence on your messenger RNA. The anticodon on the transfer RNA, you see, follows the same base pairing rules, with the A with the U and the G with the C. But the anticodon complementary base pairing would be the AAG. A pairing with the U, A pairing with the U, and G pairing with the C. So the codons are located on the messenger RNA, and the anticodons are on the transfer RNA. And the base pairing rules apply. Ribosomes are the organelles, the structures that actually assemble these amino acids to form proteins. Ribosomes consist of 2 subunits, each composed of that ribosomal RNA and proteins. The function of the ribosomes is to bring the transfer RNA bearing a specific amino acid close enough to the messenger RNA transcript to interact that permits the alignment of the anticodon and the codon. This shows the two units of a ribosome, the large unit and the small unit. So the function is going to be to bring this transfer RNA up to the, you see the binding sites on the large subunit, and where the groove at the lower between the large subunit and the small subunit, where the messenger RNA transcript will actually be delivered to. Translation consists of 3 specific stages: initiation, elongation, and termination. So RNA polymerase is going to look for a specific promoter region that's going to start. In initiation, the major players in protein synthesis come together: the messenger RNA, the transfer RNA, and ribosomal subunits. In step 1, the small ribosomal unit attaches to the messenger RNA at a start codon. And if you look at your amino acid table in your textbook, the start codon is AUG, which corresponds to methionine. In step 2, the transfer RNA with the complementary anticodon pairs with the start codon. The larger ribosomal subunit then joins the smaller one to form a functional intact ribosome with messenger RNA positioned in the groove between the two subunits. This shows just what we talked about, where the large unit, the small subunit. You see the messenger RNA transcript attached to the small subunit of the ribosome. And you see the first three sequences is the AUG designating start. So the small ribosomal subunit is going to join the mRNA at that start codon. The tRNA with the complementary anticodon is going to pair with the start codon, and on the anticodon you can see that would be UAC, complementary base pairing. So the ribosomal subunits join will form the functional ribosome. Next, elongation is going to occur as the enzymes in the ribosome link amino acids, forming a growing chain of amino acids into a protein. During elongation, codon recognition is going to occur. Peptide bonds are going to form with the previous amino acid assisted by specific enzymes. Ribosomal movement is going to occur. tRNA in the first binding site is going to leave the ribosome. The second transfer RNA is going to move into the first binding site. The appropriate transfer RNA is going to slip into the open position, and its amino acid is going to bind to the previous one. So it's just going to be kind of like just building blocks that once the one transfer RNA binds its specific amino acid, it's dropped off, translated into a protein, it will leave. The transfer RNAs are not incorporated into the protein. Once that they drop, their job of dropping the amino acid is done off, it will go out and it's called upon again because the transfer RNAs are specific. So this just shows the framework that is going to be occurring. In step 1, the transfer RNA with the appropriate anticodon is going to pair with the next codon on the messenger RNA. Enzymes are going to link these amino acids, and the transfer RNA in the first binding site is going to leave the ribosome. Again, it's not consumed into this growing protein. The ribosome is going to move along the messenger RNA, exposing the next codon. Enzymes are going to link the appropriate amino acid, and this process is repeated multiple times. A polysome is a term used to describe a cluster of ribosomes that are simultaneously translating the same messenger RNA transcript. And this is just an image where it shows that you have multiple ribosomes that may be working on the same messenger RNA transcript at one time. Termination occurs when a stop codon moves into the ribosomes. The stop codons are UAA, UAG, and UGA. The stop codon moves into the ribosomes, but there are no tRNA anticodons that pair with stop codons. So when a stop codon moves into the ribosome, protein synthesis stops. The newly synthesized protein, the mRNA strand, and the ribosomal subunit separate from one another. So the stop codon moves into the ribosome. That's the UAA designated is one of the 3 stop codons. Release factors cause the release of the newly formed polypeptide, also termed protein, and the separation of the ribosome subunits and the mRNA occur. It's easy to read the sentence at the top, the big fat dog ran. The big fat dog ran, if you notice, each of those are in 3-letter sequences. These 3 base sequences represent codons, THE, BIG, FAT. But in the sentence below it, where the E has been omitted, the sentence doesn't make sense anymore. And this is where these mutations and where errors occur. That if a mutation were to occur, you notice that everything after where that E was omitted, every letter after that has been changed. Mutations are changes in DNA, but mutations can only be passed to offspring when they occur in an egg or a sperm. Although mutations can occur in somatic cells, that affect the functioning of that cell and subsequent replicated cells. But the only way that mutations can be passed to future generations is during fertilization between the egg and the sperm. Gene mutations result from changes in the order of the nucleotides in the DNA. Chromosomal mutations occur when sections of DNA become rearranged, duplicated, or deleted. The point mutation is where one nucleotide mistakenly pairs, such as A with C instead of G. Typically, these are corrected because DNA has that proofreading ability, but as we saw in the previous sentence, a protein translated from the sequence of lettering where the E was removed would be vastly affected, would change that. There are different types of mutations, one being chromosomal mutations, and may occur when either whole sections of chromosomes are duplicated, deleted, or may be rearranged. There could be gene mutations that changes in the order of the nucleotides of the DNA. Those involving insertion or deletion of nucleotides can greatly change resulting protein and have a more serious effect. So in the top image, you see the original DNA, C with G, A with T, and T with A. But in the next image down, you see an incorrect pairing. C properly paired with G, but A improperly paired with C. So the possible outcomes is one DNA molecule is identical to the original, and the other DNA molecule now has a base pair mutation. So in the original, you see where the GUA would code for valine. In the amino sequence on the right where the mutation occurred, you see now there's a different amino acid specified, and this is where problems begin to occur, disorders, diseases that we see. Only certain genes are active in a certain type of cell. Most genes are turned off in any given cell, and this leads to specialization. Active genes produce specific proteins that determine the structure and function of a particular cell. Every nucleated cell in the human body, except gametes, the egg and the sperm, contain a complete set of identical genetic instructions for making every structure and performing every function in the body. But gene regulation occurs in that these are only going to be expressed in cells In other words, our kidney cells do not perform the same function as our liver cells. So they're going to be specialized. Genes are really just segments of DNA in patterns of A, T's, G's, and C's. RNA polymerase looks for that specific promoter region that marks the beginning of that gene to start transcription for a particular protein. Gene activity is regulated simultaneously at several levels. At the chromosomal level, in the coiling and uncoiling of DNA, when DNA is tightly coiled, the genes are not expressed. It's regulated at transcription of genes. Enhancers increase the rate of transcription of certain genes. Enhancers specify timing of expression as well. Enhancers specify genes' response to external signals and development cues such as growth hormone. Enhancers also specify the timing of the expression and a gene's response to the external and developmental cues that affect this gene expression. Steroid hormones can turn on specific genes. Steroid hormones have a lipid, they're a lipid made of cholesterol. Thus, they can diffuse into through the plasma membrane, bind to a receptor inside the cell, and actually go straight into the nucleus. They can bind to receptors in the target cell inside the cytoplasm. They find their way to the nucleus, the nuclear chromatome, and then can turn on specific genes.